years, very boring, already completed. Then you know absolute values we discussed. Then we are discussing about we discussed bone marrow aspiration, bone marrow biopsy we discussed. Then we were discussing about leukemias. So before discussing about leukemias, we are focusing on the basics of leukemia. Am I correct? Yes. So we discussed about some of the important criteria for leukemia. What is leukemia means? Cancer to the bone marrow. Okay. Aplastic anemia means what is complete dysfunction of bone marrow is called as aplastic anemia. So leukemia leads to aplastic anemia. There are some chances are there. Let us see cancers of bone marrow. That will be the leukemia, right? Leukemias. Then how leukemias are classified? Listen, this definition is very important. Based on progression of the cancer. If I came to know today my patient has cancer, it will go to deadliest form, stage 1 to stage 4 within 2 years. Okay, such cancers are called as acute cancers. Acute cancers. If they will, it will progress greater than 5 years. Such cancers are chronic. That means the progression of the cancer. Based on progression of cancer, we have acute and chronic. Then there are two series of cancers. If you remember the erythropoiesis, then you will understand. A quick erythropoiesis. 1 second. Yeah. In the erythropoiesis, you have remembered that there are two important lineages. One lineage is myeloid and other one is lymphoid, right? So we have myeloid lineages and lymphoid lineage. Lymphoid will prepare lymphocytes, whereas myeloid will prepare all up to monocytes. Okay, granulocytes and monocytes will be made by myeloid series, right? If it is an acute myeloid leukemia, okay, acute myeloid leukemia, another one is chronic myeloid leukemia, then lymphoid, acute lymphoid leukemia and chronic lymphoid leukemia. That's what we have studied so far. Then we got an interesting topic that is a teratomas. So before discussing about teratomas, I want to tell you one more thing, that is cell under stress. Cell under stress. Because stress can be anything, UV rays, microbes or anything. Cell under stress, what it will do? It will go for adaptations. Adaptations, right? Cell will try to compensate with the stress. Cell will try to cope up with the stress. So cell will adapt to the changes. Cell will adapt to the stress. What are the adaptations the cell might take? Cell size will increase, that is atrophy. Cell size might decrease, that is increased is called as hypertrophy, hypertrophy, and decreased is called as atrophy. Then what are the other things I discussed? Increased cell count. Increased cell count is called as hyperplasia. Hyperplasia. Increased cell count is called as hyperplasia. Then metaplasia. Metaplasia means one type of cell will be converted to another type of cell. That is called as metaplasia. Transformation of cell to other form is called as metaplasia. Metaplasia. Okay. Then what is the next one? Dysplasia. So, there is no proper criteria to differentiate these cells. One cell will be small, one cell will be another shape, one cell will be very large. So, there is no proper, arrangement of the cells. Such adoption is called as dysplasia. Dysplasia is a not good condition. Dysplasia might be a cause of cancers. Once the cell reaching to this stage, there is a chance of undergoing to a cancer. Cancer activity might there are chances of cancer by dysplastic stage of the cells. Okay. After dysplasia, what happens? Anaplasia. A severe form of dysplasia is anaplasia. Then that's it. The cell is out of control. Slowly cell becoming, we can say cell becoming a psycho. Okay. At the end of the day, the cell becomes so super, lost its control. Then it became a cancer. Here we can say it's a cancer, that is called as neoplasia. That's it. Neoplasia is a starting stage of cancer. Right? Neoplasia is a starting stage of cancer. Now see students. So this is the cell cycle. Okay. So half of the to the next thing. At one particular point, the cell will start dividing. Okay, the cell will start dividing, and these are the phases of cell dividing. They are, PMAT. You already know. PMAT stands for. Keep in mind, this is the mnemonic PMAT. P-M-A-T. Okay. Prophase, metaphase, anaphase, and telophase. Right. So what happens here? First, the cell chromosomes will be doubled. Okay. The cell chromosomes has been doubled in the prophase, then prometaphase. Okay. In prometaphase, there will be an appearance of spindle fibers. Polar bodies will came into the cell and polar bodies came. Then in the metaphase, these polar bodies will keep in mind. In prophase, the chromosomes will be doubled. Okay, chromosomes will be 92, 46 into 2. Okay, double the chromosomes. Is it possible to have 96 chromosome cells in the body? It is possible. That is called as cell division. In one particular phase, the cell chromosomes will be doubled. Okay, that is called as the prophase. In prophase, the cell chromosomes has been doubled. Okay, why? That is very important. Chromosome doubling is important. Why, you will understand. After chromosomes doubled, 96 chromosomes, spindle polar bodies came. Polar bodies will, see here, very interesting. All the chromosomes, 96 chromosomes, 92 chromosomes will come to the center of the cell. Very interestingly. All chromosomes will migrate to the center of the cell. Okay. After that, spindle fiber, these polar bodies will extend their spindle fibers. These are called as spindle fibers. Listen, this is a single chromosome. Chromosome has a center that is called centromere. From this centromere, we got another baby chromosomes. Okay, this is how we got 46 here, 46 here. Okay, all chromosomes has been doubled. That is what happened in the prophase. Now, all these chromosomes came into the center. Then chromosomes has been connected by the spindle fibers. These are spindle fibers. Spindle fibers will connect to the centromeres. Spindle fibers connected to the centromeres. Then the third stage, that is anaphase. In anaphase, what happens is spindle fibers will split chromosomes into two sides. 46 chromosomes will migrate to this side and 46 chromosomes will migrate to this side. Okay, after they migrated, this is the telophase. In telophase, slowly the cell cleavage will start. Okay, the cell will start cutting into two. At the this is called a cytokinesis. At the end of the day, the cell become two independent baby cells. Okay, again the cells will undergo growth stage G1, G2. Then in one phase they will start again going to the mitotic phase. That will be the splitting phase of the cells. Listen students, this will take around 48 hours to complete. I just want to tell few interesting things about cells. That's why I am telling you. This is the real-time image of cell division. This is a video took for almost 2 days. Within 2 days we got all these things. I just want to tell one interesting thing. That is here. Just imagine. This is in mitosis, 46 became 92. In mitosis, 46 became 92. Whereas in meiosis, 46 will be reduced to 23. Okay? Imagine, this is a sperm cell. This is a sperm. Okay? This is a sperm cell. It got 46 chromosomes. They are splitted into 23 here, 23 here. We got a 2 sperm cells and they will. Okay? This is a spermatozoa. Okay? The stem cell of sperms, we can say. Okay? This cell. Gonia, spermatogonia, okay? Spermatogonia will be, zoa, okay, anyway. Listen. My concept is important here. I am taking too much time here, but I will finish. We got a 223 sets of baby chromosomes. Now see, during the splitting, one abnormality happened. Assume. One chromosome having splitted properly and the entire chromosome migrated this side. Resulting in 26 chromosomes this side and 22 chromosomes this side. abnormal disjunction. Abnormal disjunction happened. Okay? Imagine if this extra chromosome is one extra X chromosome. Now this sperm has a XXY and this sperm is X0. Unfortunately, if this sperm became a baby, okay, there will be something called as Klinefelter syndrome. This is called Klinefelter. What happens is, in the women's, XX will be dominant genotype, whereas in males, XY dominant genotype. Since we got two X's in these males, they will exhibit some female characteristics because of presence of extra X chromosome. And they are called, the unfortunate people we will see on the roads who will harass us. I want to tell one interesting point here. Okay. This condition is very, very, very rare. 1 in a million can become a gay. Okay. The third person, I can say properly. Okay. 1 in a million chances or 1 in 10,000 chances. Whomsoever we are seeing on the roads, they are just doing business. I want their blood sample actually. I want to know whether they are Klinefelter syndromes or not. Okay? I was frustrated by them, especially during my train journeys to south to north. Very tough. Anyway, fine. Now, if it is not the sex chromosome, if it is some other chromosome, okay, chromosome number 22, chromosome number 22. If chromosome number 22, an excessive 22 chromosome present, their phenotype will be 47 chromosomes. All the body cells will have 47 chromosomes in this particular condition. This is called as a Down syndrome. Very, very painful. Down syndrome means the babies looks like angels. They don't know anything. They are purely mentally retarded children. They don't know anything about outside world. They will smile, they will happily play, but they don't know anything. They will become monkeys, I'm sad to say, but yeah, their behavior will be like that. Just a single chromosome, an extra chromosome leads to a Down syndrome. Keep in mind. So, we all are very blessed. All our trillions of cells has divided properly and all of us are safe. That is the conclusion I want to draw. If any such issues happen in our bodies, we might end up with their fate. We are so lucky that we are healthy, okay? Keep in mind. I just want to tell one more thing here. There are certain chromosomes that can cause cancers. If the chromosome that is dealing with bone marrow activity, when there is an issue with that chromosome, you might have a hyper bone marrow activity or decreased bone marrow activity. Either you are prone to aplastic anemias or you are prone to leukemias. So now we got one genetic reason to get a cancers. Got my point? Not only this, sometimes the chromosome might fragmented. Some portion of extra chromosome we will get, or some portion might be lost. I said how we are losing or how we are getting because of non-disjunction. Because of non-disjunction this happens and this can lead to cancers. So now you understood the reason, one of the reasons for cancers, right? So now, by that, I can say that cancers can be two types. Okay? We understood that the genetic material in the stem cells or in the baby cells, blast cells, lost their genetic control, leading to a cancer. How they lost? Are they lost because of congenital issue? From birth, they have any issues? For this, no, there is no treatment. The baby pre-exposed to this condition. There is no treatment, that's it, very sad to say, but no choice. If the genes are there, but some external virus attacked the gene, then we have a treatment. A virus deleted a chromosome, but his stem cells are healthy. In such case, a stem cell treatment will be a choice. When, yesterday someone discussing in the meeting, when there is an issue with genetics, it's very tough, okay? Is it clear? Yeah, we need to go with engineered stem cells, that is another big mystery. Anyway, then I will tell another interesting thing. You already understood what are cell adaptations, then adaptations lead to cancers, neoplasms. Neoplasms can be many types, acute, chronic, myeloid lineage and lymphoid lineages. Then I discussed about teratoma, very interesting, right? This Diksha said how teratoma occurred. Okay, this is a cancer tumor with multiple tissues. Oh, yeah. They are hungry, that's why. This is a tumor with multiple tissues. You remember, right? A tumor with eyes, organs, and I will show one more tumor. This is called as hirsutism. Hirsutism means excessive hair growth. If you identify, this is a hair in the ovary of a woman. When we did the biopsy of the woman ovary, within the ovary we got a hair. That indicates it's a pure cancer. It's a teratoma, unfortunately. So how we are getting these teratomas? What is the reason behind getting teratomas? I don't know whether I can show this picture or not, but you must understand the consequences of these things. This is a teratoma to a child. You just see the size of the tumor. The tumor is outgrown a healthy baby. Okay, very sad to see these things. These are the teratomas. We need to blame the stem cells. We need to blame the activations of the stem cells. Okay, so problems with stem cells can lead to teratoma formations. This baby won't live more than few hours. That's it. We can't do anything. Sorry. Anyway, yeah. So coming to the stem cells. I was taking too much time, but okay. See, the sperm cell. The sperm and ovum are totipotent cells. This sperm and ovum will make a blastula, a zygote. Zygote has three layers, three important layers in a zygote. They are ectoderm, mesoderm, and endoderm. These are three important cells, three important stem cells that can give, one cells will give neurons and epithelial tissue. Very interesting, keep in mind, skin cell and brain are same. Skin is just a shading of material, right? But the cell responsible for skin is the same cell responsible for neurons. That means our skins are actually smart categorized at genetic level. Genetic level skin is smarter, after brain it will be the skin who is smarter actually. Okay, that is the insights of understanding stem cells. Okay, now if there is any cancer to ectoderm, you might see a skin level issues. Okay, but you can also see some neurons in the skin. The reason is they are taking the same lineages. So yeah, by understanding the tumor, we can able to understand the type of stem cells involved, and we can go further for further analysis. So stem cell study, and this is called as the developmental embryology. There is a branch in medicine called developmental embryology where we are trying to study what cells will be connected to what cell in the body. Okay, these are the cells. Now these cells are broadly categorized into two: germline cells and vegetative cells. Germline cells means these are the cells originated from embryo, and vegetative cells are, do you believe if I say we still have stem cells in our body? Usually we will say stem cells are at beginning, during birth, during baby in the mother. We do have stem cells. One best example will be my son, okay? We all. When we are small, we have one type of tooth. They will be shedded off and we will get a second tooth, right? How we got the second tooth? That means after the first, what is the called as in English?
Temporary, permanent.
Yeah, there is one term I forgot. Premature teeth. Okay, when that premature teeth lost, there are stem cells in the gum. There are stem cells in the jaw, and those stem cells will start cracking a new teeth. Okay, now the gum cell converted to a teeth cell. That means it's calcified into a pure teeth. That is the beauty of stem cells. Understanding stem cells will have some therapeutic outcomes. Okay, they might have some answers to resolve these issues. Yeah, this is all about the stem cells, various lineages and various tissues generated. If there is a cancer to adrenal cortex, it might be a mesodermal stem cell. Better treat mesodermal stem cell or understand the pathways of these cells, and we can cure that. Cancer has answers in stem cells. When we understand stem cell, we can understand cancer. That is my conclusion. Okay. cancer and stem cells have a root connections. You already know the CML, AML, the classification. I will come here. Then this lecture will focus on the differences between. Today I will focus on myeloid leukemias. Myeloid leukemias. There are two types of myeloid leukemias: chronic myeloid leukemia, acute myeloid leukemia. Before understanding leukemias, I just want to tell you one more important basics. Yeah, you already understood how we are categorizing the cancers. You remember? First, cancer will make a small tumors. Tumors will be converted to, they will be spread to lymph nodes. Then they will start spreading to the entire body. This spreading phenomenon is called as metastasis. Now, they have categorized how much tumor it is, how many lymph nodes is spread in, how much is, length is spread, the metastatic stage. Okay, everything has their own gradings. Okay, T1, T2, T3, T0. Similarly, okay, based on the calculations of them, TNM calculation, this is called as TNM classification of cancers. Based on TNM staging, we will conclude with this one. The cancers are broadly categorized into 4 categories. They are stage 1 cancer, stage 2, 3 and 4. So stage 4 means T1, N0, 2 and M1. Okay, that means it is under metastatic stage. More than 2 nodes has been affected, and the size of the tumor is more than 2 centimeters. That is sufficient. If your tumor is, 1/10 of your pinky nail is sufficient to die. If the cancer is this much size, that is a metastatic cancer. Game over. Okay. See, if this small tumor is identified. For example, this is a breast tumor. We identified breast tumor in a circulation. Game over. That is at 4th stage of cancer. If we find a breast tumor in a blood vessel, that means it's already spread. It is in a severe metastatic stage. See, very interesting. Sometimes tumors will be in their initial stage. Okay, there is a very big tumor, but there is no nodal spreading. That is safe. The tumor is 1 kg. Tumor is very big, it is 1 kg tumor, but this tumor never migrated to lymph node or never migrated to circulation. That is a safe tumor. That is a benign cancer. This is a malignant cancer. Benign cancer will be localized. Okay, benign cancer example, a woman with a tumor in a uterus. So we will remove the uterus. It's okay, safe. We saved the woman. Okay. So yeah. Now you understood the stages of cancer also. How we categorize the stages of cancer means by understanding the TNM classification. The type of the size of tumor, number of lymph nodes and the metastasis. Is it clear? So now you understood this basics also. Then third important basics. Listen. How to detect cancers. How to diagnose cancers. Various methods of diagnosing cancers. Methods of diagnosing cancers. Okay, a general topic. Methods of diagnosing cancers. Can you tell me, students, what will be the good methods? What will be the gold standard method for diagnosing cancers? Obviously, we need to study the cells under microscope. Because cancers have cellular abnormalities, we must study cell anatomy. Cell microanatomy. That means we need to study either smears or very best will be biopsies. That is the significance of histopathology. Why we need to study histopathology? Because we need to study the cancer cells at cellular level. Because cancer cells will exhibit some phenomena like a large enlarged nucleus, cytoplasm ratios, granules in the cells. So one is a smear study cell. Biopsies. Then second one, any idea?
MRI.
Okay, scans. So you are doing MRI or radio scans, okay, CT, X-ray to see any tumors in the body. So tumors can be detected by scans. Anyone? Listen, just because you have a tumor doesn't mean a cancer. So MRIs cannot be a primary candidate to say it's a cancer. MRI will give presence of some tumor, but that should be studied under biopsy. Okay, yeah. Then next, any other techniques? Listen, Pap smears are very good. Excellent. Smears as a pap smear. In pap smear, what happens? It is a risky area. There is a high chance of getting a cervical cancer to a woman. There is a chance of getting cervical cancer. So periodically post 40 years, we will take the cervical smears and we will check the cellular anatomy. Are they under any one of this stage? The normal cell has converted to a dysplastic stage. So we are checking them and we will continuously monitor. That is one is pap smear. Anyone? Yeah. Other things I want to tell. This is a new domain to you to understand. That are every cell in our body. Every cell in our body has an antigens or markers. This term is called as markers. What is the meaning of marker? An indicator for one particular reason. Okay. For example, here you can see the difference between helper T cell and cytotoxic T cell. Morphologically, both cells are same. There is no difference between helper T cell and cytotoxic T cell. But they have a surface antigen. That surface antigen is called as cluster differentiation 4. CD4 for helper T cell, whereas CD8 for cytotoxic T cell. We need to identify the cell protein. CD4. How we can identify cell protein? Imagine this protein is an antigen. So I will add a specific antibodies that will bind to the CD4 and I will detect them, okay, by coloring or anything, okay. So with the help of antibodies, we will detect the cellular antigens. Each and every cell has their own markers. That is the conclusion. One example will be a matured red blood cell will have a surface marker called as a TER119. TER119, I don't know the full form, but yeah. TER119 is the surface marker for RBC and reticulocyte, whereas CD71 and CD44 for these stages of red blood cells. To study the stage of the red blood cell, the ideal candidates will be understanding these markers. Now we understood the significance of markers. Listen, the markers are changing depending upon the stage of the cell maturation. During a stem cell phase, they have CD markers. When they are maturing, the markers number has been increased and they change their behavior to a new marker, CAR2. Okay. CD4 helper T cell HIV virus uses a receptor called CXCR receptor. CXCR receptor is a type of receptor in helper T cell that is essential for the virus to gain entry into the cell. And we came to know there are a group of prostitutes. That's how they studied in one particular part of our world. So when they studied these prostitutes, they came to know some of these prostitutes are not getting any HIV. How is it possible? Even if they are prone to HIV infections, how these particular people, prostitutes can be a male and female. Okay. They studied these people. Why these people not getting HIV? They took their cells, they studied the markers, and they came to know they don't have CXCR marker, CXCR5 marker. Then we understood, okay, let us delete the marker, we won't get infection. Let me take a sperm cell or ovum cell. Let me identify the gene responsible for CXCR marker preparing gene. Let me remove this gene and we won't get any HIV. Yeah, genetically engineered babies who are naturally resistant to HIV. And we have a gene engineering technique called CRISPR, and unfortunately one idiot from China, I forgot his name, Leo or something, he has done this experiment on a sperm cell, and then actually they become twins. They already become adults. The experiment further, 2 twins has born with genetic mutations. He thought that CRISPR is good, CRISPR will delete the gene. Unfortunately, CRISPR has induced 100 plus mutations in their babies, and we don't know the fate of babies. It is completely incognate. No one knows what's going on with them because for their privacy concerns, no one knows. And the baby names are Naina and Laina, I guess. Yeah, no one knows what's happening, but we understood that they got many other issues because of gene editing. And who are responsible? It is, we interfere with their life. Now they are having a painful life because of our genetic engineering. So there are many ethical concerns when we are manipulating or tampering the germline cells, the cells that can give rise to a full-grown baby. So yeah, there are issues. We have the accessibility, the policies saying that you can do genetic engineering on somatic cells. That means your gum cells, cells present in the gum. But no one should touch the sperm cells, sperms and ovumes. Because sperms and ovumes can become a full-grown baby. And what is their will? They haven't done any sin, but it is because of our experimentation we made them a human guinea pigs. Okay, that is not good. Yeah. Yes. Now, methods of detection of these markers. Multiple methods are there. One method will be ELISA. Take an antibody, keep some color to the antibody, let it bind to the target marker. That is ELISA technique. Or to this antibody, if you keep any small radioisotope, that will be radioimmunoassay. Or you do one more clever technique. Take a cell, incubate it with an antibody, and let the cell pass through a laser. That will be your flow cytometric method. Through flow cytometric method, you can detect these cells. Through ELISA, you can detect these cells. Through radioimmunoassay, you can detect these cells. Then there is one interesting technique called take a tissue of the body, submerge the tissue in an antibody solution, then observe under UV light. If the antigen present, the antibody will be there and it will emit some light. This is called as immunohistochemistry. In immunohistochemistry, we are taking tissue, we are combining some antibodies, and we will visualize the antibodies. That is called as immunohistotechnique. Okay. Now, I want to tell one more term. Study protein expression in a cell. Study protein is called as phenotype studies. We will study phenotype studies, and second one, studying the cell genes. Studying the cell genes is called as genotype studies. So we will cross-verify phenotype markers with a genotype markers. Genotype means I came to know that one particular marker is missing in my patient. Maybe this resulting in cancer. Okay? Is my patient lost marker because of genetic environment or due to some tobacco or external forces? So we need to cross-verify a phenotypic expression with a genotypic expression. We will do PCR for cross-verify. PCR will be a differential diagnosis for phenotypic expressions. Okay? Now, when a patient, if it is a male, he has any prostate cancer, the best way is PSA marker. Prostate specific antigen will be a phenotype, and there are some genes, some technical term genes, and we will check those genes. That is the phenotypes and genotype expressions. So I will move forward. Specific antigen, PSA. Yes. All T cells have two types of markers. Helper T cells have CD4 marker, and cytotoxic T cell has CD8 marker. I got two same cells. On surface they are looking like same cells, but when I added CD4 antibodies, I binded with this paper. That indicates this cell is a CD4 cell. That's it. Okay. Yeah. They are also a markers. That's it. So markers can be detected by multiple techniques, as I said. Flow cytometric or anything. Yeah. Now, write down this one. So keep in mind, this you can already understand how to differentiate an anemia caused by, why we are studying in hematology, because leukemias are blood. How to distinguish an anemia from a cancer. Okay? In anemia, if you give supplementation, it will be recovered. But in cancer, that supplementation cannot work because the cells have some other problems. Vitamin B12 can cure the patient, but here vitamin B12 won't work because they are cancerous cells. Clear? Yeah, this difference table you can understand, you can study yourself. Yeah, this class is up to 12, I know. Yeah, then write down. Everyone write this table. How to differentiate an acute myeloleukemia from chronic myeloid leukemia. The first and foremost thing will be onset, rapid and acute, whereas a chronic will be slow and late. Chronic, right? Gradual and chronic. Then genetic material. Affected cells. What types of cells will affected? The cells will be myeloid, predominantly with immature forms. In acute anemia, premature cells will be more in the circulation. Premature cells will be more in the circulation in acute anemia. Whereas in chronic, granulocytes and differentiated cells will be more. Because chronic, it's taking it's been a long time, so cells are already matured. In acute, immature cells, whereas in chronic, matured cells can be seen, differentiated cells can be seen. Next one. Next one is genetic markers. These are important. Write down these genetic markers. The genetic marker for AML will be FLT3-NPM. I need at least 2 examples. That's it in the exam. You need not to by heart everything. Just remember 2 to 3 examples. Okay. The genetic marker for acute myeloid leukemia is FLT3 and NPM1. Okay, FLT3 and NPM1. FLT3 and NPM1. Whereas here the chromosomal abnormalities will be seen. In chronic myeloid leukemia, there is an abnormal chromosome called as Philadelphia chromosome. Philadelphia chromosome has a genotype called BCR-ABL1.
So CD8 and CD4 will be in the acute.
Yes, CD8 CD4, that is different. Keep in mind, a healthy cell will express CD4 and CD8, but an abnormal cell will express other markers apart from CD4 and 8. Got my point? A leukemic cell will express different markers, not a healthy markers. Healthy markers are CD4 and 8. Got my point?
Chromosome name. Name. Chromosome name.
Yeah, Philadelphia chromosome. Philadelphia chromosome. Okay, it's a type of chromosome. I will show the pictures. BCR is that chromosome mutation. Why? What mutation due to Philadelphia chromosome? Okay, that is mutation. Next one, clinical features. Obviously, in cancers there will be bleeding and anemia, fatigue, fever, infection, bleeding, anemia. Whereas here weight loss can be seen. Weight loss, splenomegaly can be seen. Chronic weight loss, splenomegaly, acute infection. Keep in mind, it is because of these symptoms we will be misdiagnosed. We will think these are anemia only. I have weight loss, maybe malnutrition. But that is not the reason. It's because of cancers, okay? Keep in mind. Then chemotherapy. I just want to tell you in a nutshell about chemotherapy. Listen, students. Since most of the acute infections are genetically exposed, okay. In acute, what we will do is, this is the bone marrow and bone. Listen very carefully. Acute fast progression. Forgot about genetic reason. They will die within 2 years. The progression is very fast. Whereas in chronic, they will die maybe after 10 years. So that is criteria to see acute and chronic. Second one. Since they will die within 2 years, the ideal approach will be, it is a bad cells in the bone marrow that are converting to cancer and that cancer killing them. I will delete the bone marrow. I will give some drugs and these drugs will destroy the bone marrow of the patient. That is called as chemotherapy. In chemotherapy, I am destroying the bone marrow, leading to complete absence of bone marrow in the body. Now that will cause another problems. What are the problems? Since there is no bone marrow, no cells in the body, you need to give blood transfusions to the patient, and the patient don't have any immune system. They became a pure, just born babies again. Even a small cold infection can kill them. So chemo are very dangerous. The patient under chemo can die because of chemo rather than cancer. So we need to mitigate the issue carefully, okay? We will keep them under chemo and we will transplant bone marrow until we are getting the right transplant donor, okay? Yeah. Then what is this chronic? Listen, chronic is very interesting. The cell division I said, right? This cell division in the telophase. Okay, in the last phase of telophase. In the telophase of the cell will be splitted. That splitting is facilitated by an enzyme called kinase. Okay, Janus kinase. Okay, Janus kinase is something. There is a enzyme responsible for cutting the cell into two cells. If I block this cell cutting, there won't be cancer. So that is the idea behind this tyrosine kinase inhibitors. Tyrosine kinase inhibitors will prevent the cell dividing. Since I prevent the cell dividing, there won't be further cancer cells. Got my point? That is the idea behind AML and CML. Yes. Yeah. In the CML, we can use safe drugs. Managing a chronic anemia is easier than acute anemia, acute cancers. Acute cancers need lethal cocktail of drugs, whereas chronic we will give slowly. Okay? The chance of survival of chronic is greater than chance of survival of acute. Okay? Yeah. Next one. I think this time is up. There are very many types of myeloid leukemias. One will be polycythemia vera. What is polycythemia means? Increased red blood cells. So that is, where red blood cells came. Red blood cells took the myeloid pathway in the erythropoiesis, myeloproliferative pathway. So polycythemia is a myeloproliferative leukemia. It can be acute myeloproliferative or chronic myeloproliferative. So polycythemia vera and others are there. This we'll see in the next lecture. Okay. Yeah, there are multiple types of anemias that we'll see. Yeah. What is your next class, students?